Hello, dear students. Sadly, we forced to move to online again, as it was. Yeah, and this is the second thing because I don't know will you ever hear me because I cannot do anything with this. It's going on second day in a row. So I just hope that you can hear me, not this bzzz. Yep. Very nice. Okay, I will try to speak a bit louder so you will hear me, not these sounds. If I will speak too loud, please tell me. I will read the chat and we'll try to adjust sound. So, today's lecture will be devoted to different in vitro systems, and the first part of this lecture will be devoted to history of cell biology, because I feel somewhat important to tell you about history. You know, science sometimes goes in circles, and when you, for example, prepare some, I don't know, articles, your papers, manuscripts, you usually make some introduction, and in this introduction you usually make some kind of preliminary data about your field, about your topic. But this data usually covers like 10 years ago, maybe 20 years ago, but not centuries ago. And these circles in science, they sometimes can be like 50 years ago, some kind of sometimes centuries. So I will give you some insight. Maybe you will find something interesting in it. And in the second part of lecture, we'll talk about different in vitro toxicity in vitro systems to assess toxicity and non-toxicity especially. And what is more important, how to pick proper models to assess non-toxicity. So, well, of course, we will start with the results of your previous test. And do not forget, we will have a test in the middle of this lecture. So, about previous results, the first question was about toxicity mechanism of crystalline nanoparticles. So, as I mentioned in the lecture, crystalline nanoparticles usually have some sharp edges, so they can definitely damage your cells by physical damage, or they can damage lysosomes or late endosomes, which have some enzymes inside. Please let me know in chat if you hear something, because if you don't, there is no point to waste my time or your time, and I will just, I don't know, switch time again, because I don't know what to do with this. I don't know even who is making this innovation and all of these bizarre sounds. So if you hear, please, you can write in chat, and I will see. So, okay, a bit of quietness. So all of other things can happen with crystalline nanoparticles, but these two types of damage are more, let's say, it's like a question about main toxicity mechanism, not just or general toxicity mechanism of nanoparticles. And about, for example, absorbance of cell crystals, there is no cell crystals. Again, some very wrong point. There is no DNA cleavage, because it's very hard for nanoparticles to get inside nucleus. For most, maybe for all even nanoparticles, it's impossible. Lipid peroxidation is just, and also generation, it's a common feature of all nanoparticles. And mitosis disruption, it's have something connected with proteins, with tubulin, for example, but not about crystalline nanoparticles. So on the stem image of the cells, what is pointed by arrows, so we cannot know for sure, but it's a matter of the question to distinguish, because it definitely some kind of nanoparticles, and if they are located inside the cell, and as mentioned in the question, TEM image of the cell, so because they're located in the cell, they will probably located in endosomes or late endosomes, because there is no other places to be located. And as you can see, they're separated from the cytoplasm, so they're in some kind of organelle. So lysosomes possess just some enzymes, and when endosome and lysosome meets, they form a late endosome or endolysosome. So it's not lysosome, just lysosome. It's definitely not a Golgi apparatus, because it have different shape. It definitely not a micronucleus, because it's located in nucleus. Not a mitochondria, and well, very funny option, it's black dots. Definitely black dots, but every joke have part of truth. So please pick variants correctly. Nanoparticles can enter the cell via each ways. So there's three correct options. It can be phagocytosis, it can be micropinocytosis, or receptor-mediated endocytosis. And I believe this year no one picked passive transport, because previous year it was so many, almost everyone picked passive transport. Passive transport only for ions, which can pass through membranes by just difference in concentration. So nanoparticles cannot enter the cell via passive transport. And through the pores, the pores are usually too small, and they are not intended to accommodate some nanoparticles. So there is also not the option. What is the primary causes of nanotoxicity in cells? Almost everything except for ATP synthase inhibition. I just take this as example, as an option, but I never see such a toxic outcome in my career or in any papers, so nope. And tubulin destruction, tubulin is a protein which involves in formation of chromosomes and involved in formation in mitosis, but also I never see a specific part of nanotoxicity, a specific type of nanotoxicity with tubulin destruction. So nope. All of other things can happen. Again, almost everyone succeeded with this question. Probably the most complicated part was to distinguish late endosomes and endolysosomes. But you need to know that late endosomes, so no, sadly I just don't have this, for God's sake. Sadly I just don't have this picture, but early endosome is when endosome just enters the cell. Then it proceeds to late endosome, and when late endosome meet lysosome, forms endolysosome. So it goes like early, late, endolysosome. That's why it even have numbers 2, 3, 4. So some of you misplaced this late endosomes and endolysosomes, but the rest of you succeeded with this question. Here you need just to remember it's three types of ROS: superoxide anion, peroxide ion, and hydroxyl radical. So all of other types are not ROS species, and some of them are not even exist, like superoxide, hydroperoxide. It's just a couple of bullshit words. So Phagocytosis, well, phagocytosis mainly for cells, but some nanoparticles can also be uptaken by phagocytosis, especially when this is not a nanoparticle, maybe a nanoparticle aggregate. So aggregates can have quite a large size, that's why they can be phagocyted. What are the main antioxidants inside the cell? So it's catalase, superoxide dismutase, and glutathione peroxidase. All of other things, well, these two are not connected with so many different sounds. So these are not connected with ROS species at all. Hydroperoxide, well, it's hydroperoxide. So these three are the correct answers. What can happen to lipids after interaction with reactive oxygen species? So it's some kind of nice words not connected with oxidation. So it's promotion, periodization, peroxisomation, and well, obviously the correct answer is peroxidation. So it's oxidation of, not even oxidation, peroxidation of lipids. About Fenton reaction, so Fenton discovered this reaction back in 19th century, so more than 100 years passed already. And Fenton reactions can lead to ferroptosis, so death because of accumulation of reacting oxygen species connected with accumulation of iron atoms, and generation of hydroxyls, because if you will look on the scheme I presented in previous lecture, it's a generation of hydroxyl, not a generation. It seems for me they make some kind of competition between two rooms, because it definitely comes from different sides now. So it's not a generation of hydrogen peroxide because it's depletion of hydrogen peroxide. Reaction between ferric or ferrous ions with hydrogen peroxide results in generation of hydroxyls. So, and it's not a catalase depletion and not a reverse osmosis. DNA damage will immediately lead to cell death? No, because cell need to understand can this damage be repaired or not. And in some cases, even if the damage cannot be repaired, cell still can survive and become malignant cell, and there can be some cancerogenesis and so on. What kind of escape you will use to survive if you enter a cell as another particle? So it's an endosomal escape. You need to escape endosome to escape some enzymes in lysosome or late endosome. So it's not all of other kind of escape, especially panic attack escape, they are not exist. The most tricky questions, but most of you succeeded. Well, this was a GIF image in your tests. So the question was mainly about example I showed you in the lecture connected with controlled damage of cell membranes with the spiky particles. So about what and how parts. What part should be and how part. For example, spiky nanoparticles, so what? Spiky nanoparticles can damage cell membranes in a controlled manner to enhance biocatalysis. So what? Nanoparticles, spiky nanoparticles, and how? By increased biocatalysis. So some of you picked the other examples, and to be honest, previous year almost everyone picked the example from the lecture, and this year almost no one picked this example and put something different. So I believe this because of offline version, because previous year it was fully online as it is now. And I always believe that offline lectures are much more efficient. So, but sadly, imagine that you are a cell, nanoparticles just damage the cytoskeleton. What function you have lost? So cytoskeleton is responsible for maintaining cell shape, and for performing some phagocytosis, because it's like fibrils that push in the cell membrane, and the cell can make some of these phagosomes and so on. Cytoskeleton is no way directly associated with ATP. Of course, if cell have some damage to cytoskeleton, it's decrease in viability at all. But it's not directly connected with synthesis of ATP or mitochondria division. There's no division of mitochondria after all. And lysate, I don't know. It's just a random variance from membrane. So cannot produce lysate. What kind of lysate? I don't know. So two correct options here. Well, what next? Just let me like here. What next? How it's called when cell compartments are dissolved in lysosomes. So it can be both called autophagy or autophagocytosis or something like this. So it's both synonyms, and all of other things are probably not exist. I do not remember about xenophagy. Maybe it's dissolving of some foreign nanoparticles or foreign bodies, but I'm not sure. So two correct options, which autophagy is more frequently used. So now we can proceed with the main part of our lecture. So what about cell cultures? For some of you, probably for all of you, cell cultures, at least term cell culture, is very familiar. But for some people who are not connected with biology in some ways, culture is just like some paintings, some connected with museums and so on. So culture in other form. But cell culture is just some kind of cells, one type of cells, usually one type, which can be maintained in some artificial conditions in controlled manner, somewhere in the laboratory, and well, this is a typical picture of some cell culture. So cell culture is just one type of cells of some origin. So it can be animal, plant, insects, or some, I don't know what else, mushrooms culture maybe. And I would want to give you some insights in history because, well, it's very interesting in some ways. So probably the first man who brings the concept of in vitro conditions. So in vitro is something happen outside, not even outside of organism, but something living outside of organism. So cell cultures are considered as in vitro systems because they are, well, some years ago, maybe even decades ago, they were in organism, but now they are already for years outside of organism and can be considered as in vitro. And probably the first person here is a French physician, Claude Bernard, who was also a founder of endocrinology, and he was the first to show, he is in the center, to show the base principles of and conditions to culture some tissues of living organism. But just look, it was like 200 years ago. So a lot of time already passed. And well, in 19th century there was not so many great discoveries, and some of them occurred only at the late 19th centuries by Wilhelm Roux, who was also a biologist, and he shown some examples of preserving living tissues outside of the body. So it was like a first in vitro systems for several days, and this is a chicken embryo on a plate. And as you can see, this is not an image, it's a drawing, because there's no way at this time to obtain some images or photos and so on. And already this year, so it's 85, it was obvious for scientists that it should be warm and some physiological solution. So it already was discovered that warm saline can help to keep the chicken embryo or neural plate of chicken embryo alive in viable state. And some other researchers were connected with works of Leo Loeb, who was American physician, and he also worked with different connectivity tissues and with serum and blood plasma, and he maintained it in blood and some tissue in viable state. So it's already like almost 20th century, and some work of, I believe it's some kind of Norwegian or Finnish maybe scientist, Ljunggren, who published this paper. As you can see it, I don't know what this language is, but he maintains some skin, some part of skin explants in viable state in acidic environment. As you can know, our skin have some acidic pH. So on our skin, it may be like 5 or 6 pH, and so at this current already at this stage of cell science, it was obvious that you should meet some criteria to maintain these explants. And probably a lot of important things was discovered on the border between 9th and 20th century. Most of them was done by a scientist called Ross Garrison, who not discovered, not invented, but very well used a thing called hanging drop technique. So for now, many things connected with hanging drops. Hanging drop, it's when you have, for example, you have some cover glass, and you can put, so if you have some, I don't know, just take a phone. So if you have some drop on the surface of a cover glass, you can put it upside down, and it will be some drop which are holding by capillary forces, and this is called hanging drop. Why it was so frequently used back 100 years ago? Because there was no other ways to make these tissues or these cells to be isolated from other surfaces. So it and also, which is probably is even more important, there was no microscopes to observe these cells. So as you can see from here, for example, it's a very simple microscope, and on previous picture, as you can see, also very simple microscope. And both of these microscopes, they're like usual direct microscope, not inverted one. And now for different cell applications, we usually use inverted microscopes. And we will talk about this a bit later, and you will understand why this hanging drop technique was used, because of some technical restrictions. So Ross Garrison used some lymphatic thrombus or lymph clot, keep this clot in form of the drop, so with a hanging drop technique. And in 1907, he observed a growth of a nerve cell, and even calculated this growth. And as you can see, this is the pictures of this nerve, and let's say the details of this picture is very nice for the paper published back in 1907, because it's also drawn in, so you need to look to your microscope and draw what you see. And considering the length of the experiment, like half an hour, maybe hours in some cases, and the growth of these neurons, it was not as easy to draw everything. So, but already more than 100 years ago, scientists succeeded in producing such an experiments. So it was some other type of experiments. For example, Barbaros in 1910s, he used a chicken plasma clot instead of lymphatic thrombus, and so on. But what happens next? It's probably one of the most important discoveries prior to discovering some first cell cultures. So Alexis Carrel, he was a French, I believe, French scientist, not even scientist, a surgeon, and he successfully used a chick embryo and some cells from this chick embryo, and well, how it will be. So he used the cells from this chick embryo to reseed it. So it was like a first cell culture. And the most important thing that this cell culture was preserved for 34 years. So I don't know for sure was there any contaminations or something like this, but it's still very fascinating how to preserve cell culture for so many years. But what's so special about Alexis Carrel? He was a surgeon, and because of this, he was very knowledgeable of aseptic matters. So he definitely knows how to preserve these cells without any contaminations. And he also won a Nobel Prize in Physiology or Medicine, and he invented the first perfusion pump. But at the same time, as many of great scientists, there cannot be only good things. So for now, some eugenic policies, especially connected with Vichy France, are considered as bad things. So despite he was a Nobel Prize winner and made a lot of things connected with cell biology, he's still known for have a leading role in eugenic policies in Vichy France. But let's go a bit further, and probably some of you working with ringer solution. So now you know how a Sydney ringer looks like, and he invented this ringer solution to maintain some life in cells. Maurice Tirot modified this ringer solution, so it also some Tirot solution, but all of these solutions, they are like maybe only salts, so it's basically saline solutions modified with in Tirot solution I believe it's some kind of amino acids, but not more. And during this years was developed trypsinization, so very important to dissolve connections between cells. And so now it was a huge step, because before the discovery of trypsinization, most of the scientists used tissues, not separate cells. And this was the problem, because tissue usually it's a bunch of different kind of cells. It's not a cell culture, it's tissue culture. And after invention of trypsinization, they can work with really cell cultures. The main problem was to pick some cells from another cells, so to separate one cell culture from the other cell cultures. Sims and Stillman in 1937, just before the Second World War, used this technique to pass some cells. And I believe during the war a lot of things was developed connected with cell biology, because there was a lot of experiments. We will not talk about these experiments here, but there was a lot of them in Japan, in Germany. But what happens, so it's original work from 1936 about trypsinization of adult tissues. And by the way, it's how it looks like back again in 1937. 37 or 36. So just look at these pictures. They looks almost the same as we can obtain now in the lab. And well, these pictures were obtained like almost 100 years ago. And we are not so far in terms of quality of the pictures. Of course, now we can even obtain some pictures of separate molecules, but on the magnification of cells, well, it's look quite nice. So probably it's some kind of tissue or even cell spheroid, I don't know, but looking quite nice for 1937. So and this is probably already after the war. And what is so special about this? Well, this is a cell culture and single isolated tissue cells. At this point of time, it called a tissue cells, not cell cultures, but it looks like a, well, normal culture, cell culture. As for me, for example, it was 1948, and well, now we came to what can be called a formation of cell biology. So George Otto Gay and Henrietta Lacks. So he was a doctor, she was a patient, and he obtained some of her cells without any permission, and the cells now known as HeLa cells. So because Henrietta Lacks. And because there was no permission, it was a lot of things connected with ethics, because it was not so good in terms of scientific ethics and so on, but still the cells cultivated to this moment. It's a lot of talks we will discuss a bit later about HeLa cells, but probably it was the first cell culture in terms of well, modern term cell culture. What's next? This man, Dr. Leonard Hayflick, still alive, still gives some lectures, and he's responsible for invention of different things, both connected with theoretical biology and some technical things. So the first thing we need to talk about, it's a Hayflick limit, so named after Hayflick. And this Hayflick limit is connected with a limit of cell division. So before all of this, before the discovery of limit by Dr. Hayflick, the cells die after some time. So some number of passages, they will die, and no one knows why. So probably conditions are good, and some of the cells can be even maintained in some blood or plasma, but they still dies. And what was the reason? The reason was that there are telomeres on the end of chromosomes, and because of shortening of these telomeres, at some point cell cannot divide anymore and it's dying. So this limit of cell divisions, which is about 50 generations, so 50 divisions, called a Hayflick limit. So this man was the first one to invent this limit, to maybe not the first to observe, but to understand why it's so important. And also one of the things is the development of inverted microscope. The first inverted microscope was developed back in 1850, but it was the first inverted microscope for use with cell cultures specifically. So before invention of inverted microscope by Leonard Hayflick, it was not so easy to observe different cell cultures, which grows, for example, in cell culture flask, like one he holding in this picture. So what's so special about HeLa cells. So HeLa cells, as I mentioned, it some kind of it telomeres on the end of chromosomes, and there's some enzyme called telomerase, and this telomerase can extend these telomeres in some controlled or uncontrolled manner. And because of high activity of this telomerase enzyme, HeLa cells considered immortal. So they can divide for just uncountable number of times. And because of this, Henrietta Lacks itself can be considered immortal because her cells are still alive and will probably alive while the humanity alive, because, well, we're still using these cells. But the question is, the question is how the cells is close to human cells. So it's already more than 60 years past, and because of these telomeres, well, everything in nature is for some purpose, and telomeres definitely for some purpose too. Because if cell will divide uncontrollably, like most of the cancer cells, they will accumulate different mutations, and these mutations lead to, well, first mutation lead to formation of cancer, in some cases, yes, and further mutations lead to formation to resistance of this cancer to, for example, against some drugs. And if you look on genome of HeLa cells, for now, they have so many mutations that some scientists, for example, Dr. Van Vellen, proposed to consider HeLa cells as a separate species. Yeah, so they are, well, like not a human after all, because of all of these mutations. But this theory was not accepted by other scientists because, well, it's still a human cells. But again, if you look on genome, the similarity with the human genome, as I remember correctly for now, is like maybe 89% or something like this. And if you look on pig genome, for example, pig are very similar to human in some aspects. Pig genome is similar to human genome for 85% maybe. So HeLa cells is more like an animal, not like a human, because humans, all of our genomes, similarity like 99%. So HeLa cells are very far from a real human. And if you want to test something on HeLa cells, it will probably have almost nothing connected with the real situation, especially when we are talking about nanoparticles. So, and that's the time. Testing time for you, and after test we will discuss a bit of things connected with, let's say, more connected with testing, not with history. But some of the history inside always also will be. So if you have some problems, put some message in chat, but I think everything should be okay. Also, if you have some questions during the lecture, you can put them in chat, and also do not forget that we will have a presentation after this lecture. The link to my Zoom you can find in schedule. Schedule can be found in Nanotoxicology folder, which was sent to you at the start of the course. And also do not forget to upload your presentations and your reports there. Now you have like 20 minutes to finish this test. Let's hurry up, it's 3 minutes. Okay, we need to proceed, so please finish your tests. I know how you like these questions with the open answer, but well, there was a lot of points after all. What's next? What's next? 1 second. Open. I'll open the chat in case you put something here. So Next, we will discussing some cell cultures from biological point, because you need to know, and you need to know that why, if we're talking about nanotoxicity. Because most of you working with nanoparticles in some ways, or at least working with something that can be tested on some cell cultures, for example, or in vitro systems. And the thing is that to pick a correct model, a proper model for your testing purposes is very important. So that's why you need to know some details about how this in vitro system in one as a cell culture works. So it can be two types of cell cultures. It's primary culture and secondary culture. Most of you, like maybe 99% of researchers, well, maybe not 99, in SCAMT maybe 99, but not everywhere, working with secondary cultures. What is primary culture? Primary culture is something that was obtained directly from an animal and having a limited lifespan. So if you take some cells just from a living organism, like biopsy for example, and maintain them in some culture media for, like mentioned here, 20 or 30 days, maybe longer, maybe not, maybe less, like 10 days, it can be called like primary culture. And in any way, this primary culture will die because of Hayflick limit, because the lifespan of cells is limited, and it's also connected with the term differentiation, which we'll discuss a bit later. Because of this, most of the research works with secondary cultures. So after primary culture, well, let's say seeded or transformed in such a way that it can be cultivated for a longer period of times, it can be called secondary culture. So most of your cultures are secondary cultures. Why, for example, using primary cultures are important? Well, if we're talking about testing of nanoparticles, sometimes it can be beneficial to understand their toxicity to use some things obtained from organism. For example, if we're talking about cancer, if we aim to treat some cancer with nanoparticles, it's a good thing to take some cells from our patient and to test our nanoparticles on these cells. Not on some cells of almost the same origin, but secondary cultures. But in this case, it's better to use primary culture. But it's not as frequently used because it's some, again, things connected with medicine, with ethics, and so on. But primary cell cultures can be used to obtain some result which will be much closer to reality than when we're using the secondary cultures. So primary culture can be in some ways even considered ex-vivo experiments, because you're taking these cells from some in vivo object. But what is the difference between other difference between secondary and primary cell cultures? I already mentioned this, that some cells can be limited and unlimited. So some cell lines or cell cultures, it's a synonym, cell line or cell culture. They can be limited or unlimited. So primary cell cultures, they are limited, and only some of the secondary cell cultures, they are unlimited. What this means? This means that they can divide unlimitedly without any signs of degeneration. And for example, HeLa cells is unlimited cell culture, but still it's not very good to maintain it for a huge amount of passages, but still. And human hepatocytes, they are limited. They can live for some days, week maybe, but they will die, and this is limited cell culture. So all of this is connected with, again, telomeres and telomerase enzyme, because telomeres on the end of chromosomes are responsible for cell division, and in unlimited cell lines there's enhanced activity of telomerase or other things which allow cells to divide unlimitedly. So another thing connected with limited and unlimited cell lines is differentiation. So we have a lot of different types of cells in our organism which are differentiated. So they have a specific role in our organism. And among these cells, for example, muscle cells, sperm cells, egg cell, immune cells, different leukocytes, macrophages, and so on, nervous cells, blood cells, bone cells, and so on. But all of them can occur from a stem cell. So stem cell is undifferentiated, and all of this is differentiated cells. And some of these cells can divide, but not all of these cells. And stem cells can divide, so they can be considered in some ways unlimited cell cultures. But most of these cells, they are limited cell cultures, so they cannot divide in normal conditions. If you artificially add some hormones, some maybe proteins, maybe if you in some way alter some gene expression in the cells, they will start to divide, but probably not. Probably not. Some cells cannot divide because of another amount of chromosomes. So what's next? It's something connected again with experimentation, and it's important to discuss because it's a matter of cell morphotypes. As you see here, cells can be of different shapes, so it's not an actual shape, it's just a scheme, but still muscle cells have some elongated shape, some immune cells have some uneven shape, so they have some pseudopodies and so on. Epithelial cells can be of cubic epithelium, can be some other type of epithelium cells. Blood cells have their own shape and so on. And that's why the cell morphologies really matters, because when you're working in lab with cell cultures, you usually see cell cultures as something like this. So it's a single cell on some substrate, usually a petri dish or culture flask, and these cells can be of different morphologies. And it can be like fibroblast-like cells with very elongated shapes. It can be epithelium-like cells with some uneven shapes, and it can be lymphoblastic cells looking like spheres. The shape of the cells depends not only on origin of the cells, so fibroblast or epithelium can be originated from skin, for example, and they are adherent cells, so they grow on substrate. They need some rigid surface to grow on, and in organism, adherent cells also form some monolayer, not even monolayers, they can form tissues. So they can adhere to each other to form some tissues. And for example, some lymphoblastic looking cells, they can be in suspension form, so they can be in blood, for example, like leukocytes, red blood cells, so they cannot form tissues. Well, blood is a tissue, obviously, but it's other kind of tissue. It's not a rigid tissue. And some cells can survive without a substrate, some cannot. So they can be adherent cells or suspension cells. And when we're talking about cell morphologists, here are some nice ones. So when we're talking about cell morphologists, here, for example, you see just one cell culture. I believe it's A505 49 cell line. And as you can see, there are 3 numbers here showing different cell morphotypes. But you can ask me why there's 3 morphotypes in one cell culture. Well, the morphotypes and cell morphology depends not only on cell type. For fuck's sake. It's something like so it depends also on cell viability. So when you add some nanoparticles or other type of toxicants drugs to your cells, they can alter your viability of your cells. So for example, cells can die or can be damaged, and because of this they can change their morphology. And for example, here you can see a normally looking spindle-like cell, so it's called a spindle-like because it's like a spindle. And for example, this cell this cell looks like a round cell, and probably this cell is dead because this cell lost its connection to substrate and just flowing in culture media. And this one, impossible, just impossible. And I live in this for a week already. Do you hear at least something? I cannot do anything with this. Sadly, I cannot say in Russian what I thinking about all of this. About all of this man, because it's so when you see some structures like this one in cell culture, it can be a matter of some degeneration due to age of this culture. So these long structures like long pseudopodias, they can be a sign of degeneration, because when we're talking about, for example, of limited lifespan of cultures, even unlimited cultures, okay, even I don't hear what I'm talking about. So when we are talking about unlimited cultures, even unlimited cultures have some limits. So they can accumulate a lot of mutations even during your passivation, when you passivate them for 50 times, for example. And if we look on this hello cells, there is a lot of these long structures. For example, here you can see these long structures, and this is not very good for cell cultures to be maintained in this at this stage. So even though you're working with a single cell line, you can observe a different cell morphotypes, and this morphology is very important when you're assessing the state of your cells, because some states can be a sign of cytotoxicity caused by nanoparticles, because we're talking about nanoparticles. Another important aspect is cell adhesion. What's so special about cell adhesion? It's I believe it's enhanced SEM image, so artificially colored, but this is actual SEM image, very nice one, and I think it's a hello cells. And as you can see, there's a lot of connections of the cells to each other and to substrate. And usually we don't see these connections because they are very, very small. So the lengths of this and width of these pillars are like less than 1 micron. And why is this important? This is important because if cell will lose this adhesion to substrate, most of the cell will die because they need this adhesion and they need these connections because, well, to maintain their life. Well, some receptors on the surface of the cell membrane connected with cytoskeleton, and it's part of a defense mechanism against cancer formation. So what I mean, as you know, cancer can produce some metastasis in some other organs. So some of the cells can detach from a solid tumor and with the bloodstream travel to another part of organism and form another tumor. So it can be called a metastasis. And our organism have a natural defensive ability to prevent this, and this ability connected with the loss of cell adhesion. So when cell loss this cell adhesion, organism understands that this cell, well, not organism, cell itself understands something going wrong. Maybe I am a malignant cell, and because of this loss of adhesion, cell can kill itself not to produce this metastasis. And most of the cancer cells inhibited this pathway of cell death to produce some metastasis. And some nanoparticles, as I mentioned already, can also cause some damage to cell adhesion. And, well, that's also important to understand. What also connected with cell adhesion? Probably I will kill someone today. What also connected with cell adhesion? It's some kind of scheme just for you to understand how it's working. So these small things here, it's these things. So they're called like, well, some kind of focal adhesion, some actin filaments and so on, collagen filaments. And these filaments on the other side, inside the cell, connected with cytoskeleton. And I already mentioned some tight junctions between the cells. So this is tight junctions between cells and cell layers. And on the opposite side, inside the cell, these tight junctions have some connection to cytoskeleton. So if you have some damage to cytoskeleton, it will definitely produce some damage to tight junctions, to adhesion to cell substrate, and all of things are interconnected. So when we are talking about adhesion between cells or cell and substrate or cytoskeleton, all of this is interconnected, and nanoparticles can damage all of these things and will cause some cytotoxicity. Another aspect which we need to discuss is mitosis. So mitosis is a process of cell division, and there's a lot of phases in mitosis. So it's a prophase, prometaphase, metaphase, and so on. And you can observe these phases using optical or fluorescent microscope, and probably some of you observed this because it's not, it's probably to obtain a similar pictures, it's not as easy, but to observe some basic patterns of mitosis, it's kind of easy task. You need just to use some actinic orange staining, and you will see some division patterns. But what's so special in understanding of cell behavior is cell cycle. Usually, when you're working with cells, you never pay attention to cell cycle. So you have some cells. You look on the cells with your eyes, with microscope, and you never think about, for example, this particular cell. Is this in phase G1, or maybe it's a G2 phase, or maybe it's undergoing mitosis. So you never think about this, and that's not quite good. Well, in general, all of the cells, most of the cells in your cell culture, somewhere in the interface. So it's interface and this mitosis phase. And as you can see, this interface is quite big, and but this interface can be divided in G1, G0, S phase, G2 phase, and mitosis itself. And why is this important? Because, for example, some of the cells which are differentiated, for example, like neuron cells, they are constantly in G0 phase. So they are doing nothing. They just living, and they not producing additional proteins. They not dividing, not multiplying their DNAs, and so on. But most of the cells which undergoes division, for example, HeLa cells or some other cancer cells, they go in constantly through this circle. And for example, after mitosis at G1 phase, cell will accumulate some of the nutrients to accumulate some nutrients to provide some energy, some sources to go to S phase. During S phase, cell can double chromosomes, and during G2 phase, which are much smaller than G1, cells again accumulate some nutrients which will be necessary for mitosis. And what are so special connected with the stages? When we're using some cell lines in some random state, all of these cells at different stages. And some nanoparticles and some drugs can act on your cells only at particular stage. For example, on the next lecture, I will show you example of anti-cancer drug called docetaxel, which can act on cells only during mitosis, because its primary mode of action connected with tubulin, and tubulin involving only in mitosis. So there is no point, for example, if all of your cells in like G0 phase, this anti-cancer drug will not affect the cells. And because of this, it's frequently used to treat some glioblastoma diseases, because most of your brain cells are in G0 phase. And that's why this drug wouldn't affect your primary brain cells and will only kill some cancer cells. But you also can synchronize your cells, and it's also a term which are known as synchronization of cell cultures. So what is synchronization? Synchronization is when all of your cells in similar phase of cell division. Why is this important? Well, this is important because you need to assess your drug on the specific phase of cell cycle. And to synchronize your cell culture, you can use a simple approach. For example, you can just grow your cells up to 100% confluence. Confluence is a term which means how many space is occupied by cells. So for example, here you see some voids, so some free spaces, and this free space is, free space. So cells occupies not everything, and for example, confluence of this sample like maybe 80%. So confluence 80% means that cells occupy only 80% of space. In case cells will occupy 100%, they will probably go to G0 phase because they don't have place to divide, because everything is occupied, and because of this they will synchronize and simultaneously goes to G0 phase. And in this case, for example, if you add some docetaxel drug to these cells, this drug wouldn't affect the cells because they are in GOZ phase and there is no mitosis going on, and there is no way to affect tubulin during mitosis, and your experiment will fail. So when working with some nanoparticles, for example, nanoparticles conjugated with some drug, you must understand how this drug or this nanoparticle works. So if these particles need to affect some part of cell metabolism, you need to understand if this part of metabolism included on this particular phase of cell cycle. So I hope this is clear, probably. But a couple of slides about history again, about cell media. But it's not only about history. I will explain. Cell media. Those of you who are working with cell cultures, and probably who are even not working with cell cultures, but still, most of you heard about cell media. So cell media, it's like EMEM, alpha-MEM, DMEM, RPMI. So most of them named after the scientists who discovered this media, like Herr Eagle, who invented EMEM, Eagle's minimal essential medium, and for example Renato Dulbecca, who modified Eagle medium. So DMEM, it's Dulbecca modified Eagle medium. Some of this, like RPMI, called after Roswell Park Memorial Institute medium, so the place where it was invented. But all of these medium are very complex in composition. And because of this complex composition, you always should be cautious about how your nanoparticles can interact with the components of this cell media. But before we proceed, just an additional slide for you about very nice thing, a teacher importance, as I called it, because it was Giuseppe Levi, Italian histologist, shown here, probably not as famous for his own deeds, because he didn't receive any significant prizes. But here are three of his students. So it's Rita Levi Montalcini, it's Salvador Luria, and Renato Dulbecco. And you see some numbers here. And these numbers, it's a year of receiving a Nobel Prize by these scientists. So I believe it's a very rare occasion in science when just one man, let's say, raised three students who received a Nobel Prize. We can just, I don't know, suggest what was the approach to teaching of this scientist, of Dr. Levy, but well, three of his students received a Nobel Prize. And what's also important, Renato Dulbecca and Salvador Luria, they both of them were virusologists, and Nobel Prizes were connected with replication of viruses. So probably some part of their work now can be connected with discovering something connected with coronavirus. So just know this scientist. But let's proceed to the MEM composition. So the MEM composition, the MEM culture media is frequently used to cultivate a lot of different cells. And this is, I believe, not a complete list of components of this media. It's a lot of inorganic salts, it's a lot of amino acids, it's a lot of vitamins and other components. And just look on how precise is composition. So it's a micrograms in some, just look on this number. So it's a very, very precise composition. And why is this fascinating? Because when we are looking back in the start of cell biology, people just using some blood plasma, some lymph clots to cultivate some cells, and they, well, succeeded in some ways. But now, to successfully cultivate some cells, we're using so complex composition, so complex medium, and still sometimes we cannot succeed. But the question connected with nanotoxicology, not with this complex composition, but just look on how many things again in this, and most of these things can somewhere interact with nanoparticles. What this means? It means, for example, two options, at least two options. Option one, some of your nanoparticles, or maybe all of the nanoparticles, can adsorb on their surface some of the nutrients, for example, some inorganic salts, or maybe some amino acids. And because of this adsorption, it will be a depletion of these amino acids in culture media. And because of this, some cells will not receive enough nutrients for cell growth and will, well, in the worst case, they will die. Why is this? Well, this is bad, but why is this bad for you as a scientist? Just imagine, you add some nanoparticle to your culture media and cells died. What will be your first thought? Well, these nanoparticles probably toxic. But it can be opposite case. These nanoparticles can be completely non-toxic. But because they adsorb some part of nutrients from culture media, the cells died just because of depletion of nutrients in culture media. And, well, it's quite a complicated matter to discuss. But you always should perform, and on the next lecture I will show you some examples, you always should perform not one, not even two, but at least three or even a larger amount of tests to understand what is the real cause of cytotoxicity of your nanoparticles. Second option, which is connected with composition of media, is connected with again adsorbents on the surface of nanoparticles, but this time nanoparticle will enter the cell. What will happen? For example, the cell need, let's say, for example, some some amino acid, like again glutamine, valine, yeah, and cell need just 1 nanogram of this acid, just some random numbers. Cell need just 1 nanogram, but with this nanoparticle, with adsorbed amino acid on the surface, let's say 100 nanograms enter the cell. What will happen to cell? No one knows, because it's completely can be completely random damage. It's the same thing when you, for example, mistake some drugs. Instead of one drug, or instead of one pill, you picked 100 pills. Where you will be after taking 100 pills? In a good variant, you will be in the hospital. In a bad variant, well, everyone knows. So your nanoparticles can be completely non-toxic, but because of their natural ability to adsorb something on the surface, or in case you have some porous nanoparticles, they can even absorb in the whole volume. They can bring some unnecessary, some extra amount of nutrients inside the cells and cause some damage. So because of this, it's important to study the behavior of your nanoparticles not only in buffer solution, for example, aggregational stability or sedimentation stability, but it's also important to study some interaction of culture media with your nanoparticles. because it can give you some answers to cytotoxicity effects. So I believe, well, it's still a lot left to discuss. Still a lot, by the way, and we already like 5 o'clock. I will try to be a bit faster. So the main stages of working with cells, not very important for all of you, but why is this important? We can just skip this one, it's just for you to understand that there's a lot of stages of working with cells. You need to obtain them from some primary culture, from cryobank, you need to passivate these cells, you need to produce, to maintain, perform some experimenting, and again put these cells in some cryo boxes and so on. But for you, mainly as a chemists or some researchers who working with nanoparticles, the most important question is about picking a right model for testing. Why is this important? For example, just imagine you synthesize some nanoparticles. It doesn't matter, Hafnium nanoparticles, magnetite, chitosan nanoparticles, and you want to test their cytotoxicity. And you come to a cell biologist and with some kind of nanoparticles, yes, and ask to test these nanoparticles. And now it's up to cell biologists, not up to you, to choose a proper model for testing. And in 90% of cases, the test model will be we have now this cell culture in vogue, so we will test on this culture. And no one will ask you about, do you want to test on primary or secondary cultures, limited or unlimited, normal or malignant, transformed, not transformed, in which stage of cell cycle. Yeah, I remember 5 minutes. 5 minutes before presentation, we will postpone a bit. So no one will ask even about what kind of tissue you want to test your nanoparticles. And all of this is very important because picking a right model, well, it's important for outcome. And I will show you some examples. Some of the slides already was shown to you, but let's just discuss in a bit of details. So all of this is connected with cell cultures, and cell cultures, as I mentioned, can be adherent or suspension cell culture. So adherent is a monolayer, so it's a 2D cell culture, some on the surface. Suspension cell culture, it's in volume, but it's separate cells. But it also can be some in vitro models in form of 3D cell cultures, also called as cell spheroids or organoids. What are so special about these cell spheroids? So when you have some suspension cell culture, you can form some cell spheroid with a hanging drop technique or using some polymers and form some nicely looking spheroids. This is some example from, I believe, some Nature paper. This is example from our laboratory. And what's so special about cell spheroids? In our organism, very little number of cells form just a monolayer. So it probably may be endothelial cells, maybe some other cells, some basement membranes and so on. But in most of the cases, cells in our organism forms tissues, and these tissues have some volume. And because of this volume, there's gradient of cells, there's gradient of nutrients, and so on. And when you're testing some nanoparticles on just a monolayer cell cultures, it's not very close to a real situation in organisms. That's why you need some 3D cell cultures. And what you can study using these 3D cell cultures? Well, you can study, it's just some microfluidic device. And this is example, for example, of not even a 3D cell culture, it's a Also connected with this, but still, next step, organ on chip. So also can be considered as 3D cell culture or 3D cell models. So you can, for example, produce some cell layers like this one, cells of one type, cells of another type, and you can study some intra-layer transport of nanoparticles or other drugs. Because, for example, if you study some nanoparticles which are intended to be used as inhalation, so you inhale some nanoparticles, and if you want you want to study will these nanoparticles pass through lungs to, for example, to blood, and you build some system with lung cells and epithelial cells and study this border. So it's quite useful for some advanced studies, not for basics eliminations, not for basic manifestations of cytotoxicity. What can be other types of in vitro systems? Because today we're talking about in vitro systems. Other types of in vitro systems can be artificial systems. So artificial systems are quite far from cell cultures, even from 3D cell cultures, and they are like a next step in lab on a chip. So here are some examples of artificial veins or arteries, probably artificial circulatory system, and this is artificial valve from a heart. And why you need to such a system to test something? Because, for example, if you aim to produce some nanoparticles which can affect or interact with, let's say, some walls of your veins, probably you need to produce something which at least looks like a vein. Because it's important to consider some shear stress, some velocity. Don't know where to put it. So it's important to consider some velocity of blood and a lot of other things, because, well, it's important. I'm waiting for this. But also on the very first lecture I give you a very short example about X-Vivo systems, and I need to add just a bit of information why this X-Vivo system can be useful for studying something connected with nanoparticles. So nanoparticles is very small, it's obvious, and when we working with separate cells, it's not information obtained just with cells, just with cell cultures can be a full information, because it's just a cell cultures without some surrounding, and they behave not like a real organism. When we test some of the nanoparticles in the real organism, like in mice, for example, it's quite hard to observe these nanoparticles in living organism without killing this organism. For example, we need to know how nanoparticles transfer from one organ to another organ, or biodistribution of these nanoparticles in real time. In some cases, it is important to observe something in real time, and in living organism, it's not you cannot do this, or only with a very complex equipment. But if you have some separated organs, for example, like shown here, lungs, or even heart or brain or stomach, you can study more precisely how nanoparticles interact, how, in most of the cases, this X-Vivo very complex system, in some cases, X-Vivo system is more complicated and more expensive than in vivo systems. Because in vivo system is just take mice, kill mice, obtain some liver samples and study, make some histology. And as you can see here, it's some kind of bioreactor with the lungs. It's a peristaltic pump. It's some kind of pressure should be maintained here, some ventilation. and look like this one. It's, I believe, some stomach on chip or something like this. So it's some skin explants on a specific plate with some electrodes, which can simulate some nerves and so on. So in some cases, ex-vivo systems, they are very complex, but at the same time, they allow us to discover some things which are impossible to discover in living organism or in vitro systems. So they are quite useful for some specific, very narrow, very specific approaches. And well, finally, there are some examples. Some of these slides already presented in previous lectures. But now, why I want to return to this? Because it's examples of raw models for cytotoxicity assessment. Again, it's just my opinion, but I believe the scientists, the researchers in these papers could pick a more proper model for their study. For example, this one, a hemopolymer, which is used for dental fillings in our teeth. And the toxicity of this polymer was studied on human lung epithelial cells, BS2B. The question is, why polymer intended for use in our mouth tested on lung cells? Well, yes, it's still epithelial cells. But the only reason for this is that probably some part of these fillings with the air can come to our lungs and we can breathe this hemopolymer. But I believe the amount of this breathed polymer is so small it cannot affect any of our epithelial cells. But why not to take some cells from actual mouse? So some human mouse epithelial cells, I don't know. Probably because this was the closest one available in laboratory. But it's obviously not the best option. It may be a good option, but not the best. Another one example, brain delivery with some lipid nanoparticles, chitosan, again, brain delivery. But where they studied this brain delivery? A549. What is A549? It's cancer cells from lungs. So again, brain delivery studying on cancer cells from lungs by MTT assay, and MTT is not a best option again. Of course, they studied some histology samples from nasal mucosa because these chitosan nanoparticles intended to be used as inhalation nanoparticles, and also, which is very suspicious, they tested these nanoparticles for fetal toxicity, and it's not, well, let's say there's a lot of common tests to test and the toxicity of your nanoparticles, but fetal toxicity, it's not very common. So probably they knew something about their nanoparticles, so they picked this specific test for fetal toxicity. But still, in this paper, probably it's not the best journal, of course, but still, nanoparticle tested for brain delivery, not tested for cytotoxicity on brain cells. Another example again, I believe it's again chitosan nanoparticles and chitosan oligosaccharide coated-iron oxide nanoparticles. Toxicology reports do not remember, probably not also not very good journal, so you should not repeat their mistakes. Look at these cells. It's a HeLa cells, again for brain delivery. A lot of chitosan nanoparticles used for brain delivery. HeLa cells, I already talked a lot about HeLa cells today. It's not a human, after all. Again, lung cells and HEC 293. HEC cells, it's partially endothelial, partially neuron cells from kidneys. Where's kidneys and where's brain? Where's brain and where's lungs? And HeLa is not human. So they tested particles for brain delivery on cells from not even our nose, so it's not even epithelial cells from nose, not some neurons. Well, here can be a bit considered as neurons, but not from brain. From a kidney, it's partially undifferentiated cells. Well, if we're looking on some conclusion from this paper, as you can see here, we want the aim of the present want to investigate condition can be devised where pegylation of chitosan would reduce toxicity and so on. But toxicity, it's another work here, and pegylation of chitosan would reduce toxicity towards nasal mucosa while maintaining ability. So people talking about reducing toxicity to nasal mucosa cells with pegylation of chitosan nanoparticles, and really pegylation helps to reduce cytotoxicity. But look at the cells. It's a callus cells or lung cancer. Yes, it's epithelial cells, but it's not the cells of nasal mucosa. How can you say about reducing toxicity to nasal mucosa? You test your nanoparticles on lung cancer. With the same option, you can test on epithelium obtained from intestines or stomach, for example, or kidneys. So for me, there is no point if you really aim for some medical application. If this paper just to publish, let's say, work just to publish, to obtain some paper, okay, you can test on anything you want. But if you really aim to some medical application, you need to pick up a proper model. And well, all of this works without a proper models. And the last slide, I believe here, is about testing cycle. So, how many systems we have? We have first some, well, when you obtain some nanoparticles. Let's talk about nanoparticles. After all, so if we are talking about nanoparticles, after you just obtain these nanoparticles, you should perform some preliminary testing just to understand a general patterns of cytotoxicity. And for this purpose, you can use some unspecific cell lines, even cancer cell lines, because they possess still a lot of similarities with our normal cells. And for general testing, it's okay to use some unspecific cell lines. But if for unspecific cell line your nanoparticle is non-toxic, you need to go further to some specific cell lines. For example, this one is HUVEC cell line, so it's human vein endothelial cells. It's endothelial cells of our veins. And if you, for example, drug intended to be used intravenously, you obviously need to test its toxicity, cytotoxicity, on this specific cell line. The question is that this cell line is very tricky to cultivate, not very easy to obtain and maintain it live. Because of this, most of the researchers skip this stage and use some other not endothelial epithelial cells, like lung epithelial cells. But the answer to the question, is these nanoparticles will be toxic to your endothelial cells, well, remain unanswered. After you perform some experiment on specific cell lines, you should move to advanced in vitro models. For example, artificial veins or 3D spheroids or some other artificial things. So it's up to you. It's a lot of things can be considered as artificial in vitro models. After this, you can probably go to X-Vivo models. So this is an example of blood clot obtained from a patient. And for example, if you go to dissolve this blood clot with your nanoparticles, you should probably take one blood clot and try to dissolve it in some ex vivo conditions. And if you succeeded, finally you can go to in vivo models with some animals. So this is some example of a rat. How it's called? Forget how it's called, but it's the things which hold intestines together. Very nice model for testing some nanoparticles and some blood coagulation, because it's very easy to investigate under optical microscope. So, but apart from the first one, apart from unspecific cell lines, all of these stages requires a lot of money, a lot of expertise, and a lot of specific equipment. And because of this, well, probably the less expensive is the first one, and well, everything connected with the cells not very expensive. But if you have some animals in your institute, in your laboratory, usually it's not very hard to perform some basic experiments on these animal models. So in some papers you can even see like researchers skip everything between unspecific cell lines and in vivo models. Sorry. So, and probably that's all. All for today. If you have some questions, you can ask me in chat, and do not forget that probably like in 10 minutes we will start our session in Zoom. So today 2 people should present their topics. It's Elizaveta and Paulina, and all of other people are also invited, just to see how it will go on. A link to Zoom was sent to you yesterday, and also you can find this link in your schedule. So if there is no question, thank you for attending this lecture today. We'll see you tomorrow, same time. Bye bye.